We've been around the part of the University of Arizona since '97. We're an integrative group, originally psychology, mathematics, philosophy, and anesthesiology. I'm in anesthesiology. The other departments, Al Kazniak was the first director, then David Chalmers, who was a philosopher. And we were funded originally by a grant from the Fetzer Institute. And the Fetzer grant ran out, and Dave left about the same time. And I became director. And since then, with the capable management of Abby Behar Montefiore, who's sitting in the back, we've managed to survive just based on our conferences that we put on. We put on the Tucson Conference every 2 years toward the science of consciousness in even years. And in odd number years, we have a conference elsewhere in the world. And the next one will be here in Stockholm, in Stockholm, Sweden, May 2 through 8th, 2011. It should be very, very interesting, and I encourage all of you to come to that. And there's plenty of information on our website. So the purpose of this seminar is about microtubules, and this is an image from Anirban Bandyapati. He'll be the third speaker, and he'll explain what this is. We're going to cover several topics, and I'll set the stage why this is relevant, why I think it's relevant, about megahertz coherence, ballistic inductance, and the possibility of quantum computation in microtubules. And after me, Jiri Pokorny from the Czech Academy of Sciences and Anirban Bandyapati from National Institute of Material Sciences in Tsukuba, Japan, will speak. And we all three spoke at a Google workshop on quantum biology last week. When was that? Just a couple days ago, actually. And then also I guess that was last Friday, and then we've been in a workshop on quantum biology and cancer at Arizona State University. So the possible role, or the role of quantum physics in biology has been growing. It was thought of as sort of a very odd idea by when it was first proposed by some of us way back when, but it seems to be catching on, and more evidence has come forth. Mainly about 6 years ago in the area of photosynthesis. It turns out when plants absorb photons, the energy is transported to another place for chemical energy, and the energy is transported in a way that takes advantage of quantum coherence. That is, the energy, electrons or photons, occupy all possible pathways. This is why plants are relatively efficient at converting photons to chemical energy, which provides food and basically life on Earth. So my feeling is if a potato can utilize quantum coherence, our brain probably has figured out a way. But again, proving it is difficult. So this is an immunofluorescence of a double nucleated cell. And the green are microtubules, which form part of the cytoskeleton. They are the rigid components of the cytoskeleton. They self-assemble. And the more asymmetrical a cell, the more it needs microtubules. A spherical cell or a bacteria with a stiff cell wall doesn't need microtubules, but they form the skeleton, the bone-like scaffolding within all animal cells. And a number of us for many years have thought that they not only are they the bone-like skeletal support, but also are the sort of the nervous system of each cell, providing sort of the real-time onboard computer for what's going on inside the cell. In mitosis, the blue are the chromosomes, the red are the microtubules, and the mitotic spindle microtubules grab the chromosomes, separate them into daughter cells, and if this doesn't go absolutely perfectly, if there's an abnormal separation, you get abnormal genotypes and cancer. And a theory about 100 years ago by a German biologist named Bovary said the problem in cancer is abnormal mitosis. That is, if the chromosomes are divided not quite perfectly, you're going to get an abnormal genotype, some of which are going to be non-receptive to feedback and go on to form malignancy. Now, genes came along in mutations, and all the attention got put on mutations, oncogenes, repressor genes, and stuff like that. But that really hasn't borne a lot of fruit, and now there's at least in some circles, a move back towards examining mitosis. And when I was in medical school about 40 years ago, I spent a summer research elective in a cancer lab and got interested in mitosis and how these microtubules knew where to go and what to do and how they were organized. They seemed to have some intelligence inside the cell. And at that time, it was also discovered that microtubules were in neurons. prior to 1972, believe it or not, the fixative agent for electron microscopy was dissolving all of the microtubules. So the interior of cells under electron microscope looked like a watery soup, just some things floating around with no structural organization whatsoever. But then a guy named Keith Porter switched from osmium tetroxide to glutaraldehyde, and all of a sudden saw all this cytoskeletal structure, including neurons. So my real interest was in the brain and neurons, and so I got hooked on microtubules and tried to figure out how they seem to have intelligent capabilities. At the same time, computers were just becoming in vogue, and I looked at Boolean switching matrices and how computers operated, and began to make models and metaphors of microtubules as information processing devices. So this is more mitosis. In this case, the chromosomes are in blue and the microtubules in red, and the yellow are the centrioles. And centrioles, I'm going to digress just a minute because they're very interesting. They're actually made of microtubules. So each of these cylinders here would be one microtubule, and 3 of them, or sometimes 2 in different structures, fuse longitudinally, and then 9 of those triplets form this megacylinder. And this is the same structure as in cilia, flagella, except there they have central pairs with motor proteins that bend. These guys are just the navigators and the pilots of mitosis, and also perform other functions that we probably don't have a clue what they do. They're always in this very odd perpendicular arrangement with the 2 megacylinders, and the 2 together make a centriole. And interestingly, the centriole and cilia and flagella are the exact precise size to be optical waveguides. They're the size of a photon, if you will. And some people have suggested that they are quantum optical devices, and there may be quantum entanglement between centrioles and cell division. And in fact, Albrecht Buehler at Northwestern for many years has argued that the centriole is the intelligence, and the microtubules are the intelligence inside the cell. And he's done this and many other beautiful experiments where a piece of a cell, he's cut out the nucleus, he's cut out everything except the centriole and a bit of the cytoskeleton, the microtubules, and when he shines a light, the snippet of the cell, it's not even a complete cell, moves towards the light. And he showed conclusively that it's the centriole that detects light. The same structure is in our retinas and in all primitive optical systems, and so these centrioles or cilia may be some kind of optical detectors. But that's a bit of digression. So let's go back to single individual microtubules, for example, inside neurons. I'm often amused at people who say that the brain is 100 billion simple switches, with each neuron being an on-off switch, and yet a single-cell paramecium can swim around, find food, find mates, have sex, can learn. It doesn't have any synapses at all. It does so using its microtubules, its cilia, and its internal microtubules. So I think to understand the problem of consciousness, which is my main interest, we need to look inside neurons to the level of microtubules. And as we see here, in this work from Nancy Wolf, here are 2 synapses on the left. At the top we have a dendritic spine, and on the bottom we have an acetylcholine just synapsing on the dendrite, and the receptors and so forth. And inside we see this network of microtubules. And if you look carefully, you can see that they're in different polarity. This one is kind of going this way, and this one is kind of going this way. And as we'll see later, microtubules have a polarity on 2 different ends. And in all cells, except for nerve cells, they are radial. The same end is at by the nucleus and the centrioles, and pointing outward in the same direction, like spokes on a wheel. And that's true in axons. In dendrites of neurons, they're interrupt and they're continuous. They just go pretty much from the center of the cell out to the periphery. However, in dendrites, they're interrupted and short, as we see here. They're in mixed polarity, and they're interconnected by these microtubule-associated proteins, which some of us believe process information, including storing memory. And this is like a network, an information network inside the cell processing information. Now, that's been a theoretical idea that's been around for a while, and with some assumptions attached to it. Now, we know that microtubules are important in the nervous system because if they go bad, you get Alzheimer's disease. Now, Alzheimer's has 2 lesions, there's the amyloid plaques, which get most of the attention and the research money for some reason. And here's normal neurons. But inside the cell, inside the neuron, are neurofibrillary tangles, which are actually microtubules that have are disintegrating. So here's a healthy neuron. Here's a diseased neuron, Alzheimer's neuron. You can see it's kind of crinkled on the outside. And if we look at the microtubules inside, we see that they're kind of they're messed up. And they're messed up because tau, which is a protein, a microtubule-associated protein, is hyperphosphorylated. It falls off of the microtubule, and the microtubule disintegrates, and you get cognitive dysfunction. You lose memory and eventually consciousness. And heavy metals poison microtubules and give a picture very similar to Alzheimer's. So here's a close-up of that. And now what tau does is an interesting question, because it was always thought to be, as you see in this picture, something that kind of knits together the microtubules, binds them together, prevent them from falling apart. But actually it turns out that they're more important than that. And recent studies show that tau acts as traffic signals for motor protein transport of synaptic materials. So for example, if we have these motor proteins, kinesin and dynein, which move along microtubules, they actually kind of prance. They actually kind of jump and leap very efficiently somehow. And remember that the microtubules are interrupted, and in a neuron they're branching, and they actually to get to a specific synapse downstream or upstream, depending on how you're looking at it, up in a dendritic arborization, some dendritic branch, they have to make choices and switch back and forth, and they have to know when to get off the microtubule to deliver their precursor, the precursor of the synapse or the enzyme, whatever it is, to the right place at the right time in demand of the synaptic plasticity. This is all part of synaptic plasticity, which is the cornerstone of learning. Well, anyway, it's now been shown that tau is a traffic signal, and for example, tells this kinesin where to get off, where to depart the microtubule and deposit its cargo, whatever it happens to be carrying, to a particular synapse at a particular area. So then the question is, how does the tau, so the exact placement of the tau proteins on the microtubule then determine where these precursors or these enzymes and receptor components are going to be delivered. And you could say, well, tau's really smart, it knows where to go, but that doesn't really make any sense. The other possibility is that there's information embedded in the microtubule, which is, after all, a lattice and has a capability for information processing, and that basically codes for where the tau should bind, and that in turn codes for where the enzyme precursors and synaptic precursors should be dropped off and delivered. Now I want to mention one other recent finding that relates to learning and memory in microtubules. Long-term potentiation is a model for learning that's been around for many years. It's in vivo mainly, but also in vitro in virtually any synapse. If you have rapid presynaptic inhibition, for a short period of time, high-frequency stimulation, and then stop it, there's an enhanced sensitivity at the postsynaptic receptor. That synapse becomes more sensitive to a given stimulus, and this is thought to be the paradigm for learning. And that synapse is reduced, has enhanced sensitivity, reduced threshold. But the precursors or the materials that make up synapses, as I showed you in the previous slide, are very transient. They last hours to days, and yet memories can last a lifetime. So memory must be somehow hardwired, somehow stored in the neuron or somewhere over a long period of time. And this remains unknown, remains a mystery. Now, in the last few years, it's been discovered that this CAMK2, it's called, which is a hexagonal snowflake-shaped holoenzyme, is an important mediator between the synaptic events and long-term storage of memory. And in fact, when calcium comes in through these receptors, glutamate or other neurotransmitter receptors, it causes calcium calmodulin to assemble and form a holoenzyme in this hexagonal shape. And that in turn binds with gets with the microtubules and is distributed throughout the dendrites very rapidly, and that correlates with learning. So a number of us got the idea that CAMK2, the question is, and CAMK2 phosphorylates a substrate. It's known to be able to phosphorylate various substrates, and that somehow correlates with memory. So the question is, where is the phosphorylation occurring? What does CAMK2 phosphorylate that stores memory, at least short term, at least for short-term memory, medium-term memory, if not long-term memory? Well, here's a CAMK2. This is what it looks like after it's been assembled due to calcium influx. And with more stimulation and phosphorylation, the CAMK2 transforms, much like those transformers in the movies, the cartoons, or whatever they are. And actually, these 6 legs, or arms, depending on how you look at it, extend above and below the middle section, which is called the association domain. And these are the kinase domains. These domains, the 6 of them above and below, are able to phosphorylate a substrate, and that encodes memory somehow. And here you see the phosphorylation sites. They're actually on the upper side of the feet, if you want to call these feet. This is kind of like an insect-looking thing, or some people call it a nanopoodle. And I like that. And the phosphorylation is on the upper side. So the question is, what is phosphorylated by this guy to store memory that can be utilized in the cell? Well, as you might suspect, I think it's microtubules. And so I had this idea, and I went to my colleagues Jack Tuszynski and Travis Craddock at University of Alberta in Edmonton, and they are both also microtubule fanatics, I might say, along with me and a few other people here. And I told them of the problem, and they do molecular simulation. And so they started, Travis did the previous slide I just showed you of the CAMK2 activating. And here's a microtubule here. It's hollow. It's made up of these 13, well, you can't really see that there's 13. There's 13 of these protofilaments. You can see that it's hollow looking at it on end. Here it is on the side view. And if we look at a close-up at 5 nanometers here, these are the building blocks. This is tubulin. Tubulin is a peanut-shaped protein with an alpha and beta end, and these C-termini tails that stick out into the cytoplasm, which is why it kind of looks hairy here. And these are negatively charged when they attract positive charges, so that there's a counter ion layer, a double Debye layer on around surrounding microtubules, which might protect it from decoherence for quantum effects, which we'll come to later. Anyway, these are the phosphorylation sites on the tubulin. This is where tubulin can be phosphorylated and could receive phosphorylation energy, which might store memory. So the question is, how does the CAMK2 relate to this in terms of geometry and size? It turns out it fits absolutely perfectly. Here's this is CAMK2, as we saw earlier, where we've taken off the top so we can just see the lower set of 6 legs. So each of these kinases can phosphorylate a site or not. So you can think of it as a bit of information. So each CAMK2 conveys potentially 6 bits of information. Now, if we overlie the CAMK, here's a lattice and a B lattice. So microtubules can be in these 2 different lattices. If we overlie the CAMK2 on the A lattice and the B lattice, we see that it matches perfectly, allowing a little bit of flexibility in the extenders that I showed you earlier. So the geometry and the size are precisely perfect, and we know from other work that microtubules are, that CAMK2 binds to and is actually moves along microtubules. So here's an image of the nanopoodle, if you will, binding to, sitting on, maybe even moving along by hydrophobic interactions, a microtubule, and depositing information potentially by phosphorylating the substrate that it's sitting on. And here we show, this paper is coming out in the Journal of Integrative Neuroscience any day now, and we show that the phosphorylation mechanism can occur either through the C-termini tail or by this thing unfurling, and we show the specific amino acids involved in the phosphorylation. So the point is this, that if we take a microtubule A lattice here and here, or a B lattice, and make some assumptions about the the phosphorylation, whether it can be on just the on either the alpha or beta tubulin or both or either, then we can see that the information capacity from one CAMK2 through synaptic events depositing information on a microtubule is vast. It's enormous. So the simplest case would be 2 to the 64, 2 to the 6 possible configurations. We show 6 out of 64 here. If we assume 3 states in an A lattice, because either the alpha or the beta can be phosphorylated, we get something like 500 possibilities, and this is just from one CAMK2. And if we assume the B lattice, where it can orient this way or that way, it's something like 5,200 possibility bits per byte. Now, an ordered array of bits is a byte in computer terminology, so we call these memory bytes conveyed by CAMK2 onto microtubules. So this is supportive evidence for the possibility that microtubules might be processing information in some way. And actually, that's a fairly old idea, back to the early 80s at least, where the basic idea is shown here. Here's a microtubule, and if you take each of its subunits, it can exist in 2 possible states, and possibly a quantum superposition of both states, which would make it a quantum computer. Now, if you look at the lattice like this and just define a neighborhood patch and calculate the dipole interactions, because each of these has a dipole, and there's dipole-dipole coupling among neighbors, you can, and then you can play a sort of game of life, cellular automata. Now, the game of life is shown here. It's the simplest form of computation, but can actually be very, very complicated. If you have a simple orthogonal checkerboard and each cell, each unit can be on or off, dead or alive in the game of life, and make some simple rules like if you have one neighbor or 3 neighbors or 1 neighbor or 4, 0, 1, or 4, you either have not enough or too many neighbors and you die the next generation. If you have 2 or 3, you live, and you make these simple rules and then have a clocking mechanism so that at the same time each of them calculates their neighbor interactions. At the next generation, you have a new set of states. And this is basically a cellular automata, the simplest form of computer. And if you do that in the game of life, you get patterns and gliders that move through this lattice and can solve simple problems, or actually if you have a large cellular automata, can solve differential equations and all kinds of stuff. So anyway, here's this type of molecular automata on a microtubule showing patterns moving through a microtubule. And this raises the potential capacity in the brain enormously, because the number of bits in a given neuron is vast. Now bits are not simple, neurons are not simple switches, but they're now very complex computational devices if we consider the microtubules are the computers. But this depends on a number of things, including a clocking frequency. And for this, so here's, for example, 1 bit of microtubule with patterns evolving over time. Now, the time steps, I did this work in the late 80s and 90s with some physicists, and we assumed the clocking mechanism went by a mechanism proposed by a guy named Herbert Frohlich. Frohlich Coherence or Frohlich excitations. Frohlich was a German physicist who worked on the theory of superconductivity and then switched to biology. And he came up with this idea of what's now known as Frohlich coherence or Frohlich condensation or Frohlich excitations. And the basic idea was that if you have a dipole, and he originally assumed it was membrane proteins in a membrane which has a gradient, that if you supply energy, non-coherent energy, random energy, even heat from the heat bath, that these dipoles would couple to each other and oscillate coherently, so that you would get coherent excitations, something like a laser. A laser takes non-coherent energy and converts it to coherent vibrations. And he said this can happen in biology. And he applied it to membrane proteins, and I asked him personally, because I met him, whether microtubules would qualify, and he said, well, if they have dipoles in each tubulin, then yes. And so we began using the Frohlich coherence as a mechanism to clock our molecular microtubule automata. And we're going to hear more about Frohlich condensation from Professor Pokorny in a moment. So subsequent to that, I ran into a stumbling block in terms of microtubules solving the problem of consciousness, because people said, well, let's say you're right and you have all this information going on inside the neuron, and there's vastly more information than we think. How would that explain why we have feelings, joy, pain, conscious awareness? And this question stumped me, and it later became known as the hard problem. It's been around forever, but the question of why we have conscious experience as opposed to just cognition and non-conscious behavior. And fortunately at that time I read a book by Sir Roger Penrose, who was arguing for quantum physics involved in consciousness, a type of quantum collapse. But he didn't have a good quantum computer in the brain. He didn't have a good qubit, it's called. A bit would be something that could be 1 or 0. A quantum bit or qubit would be something that could be in both states at the same time. So he thought maybe that neurons both fired and didn't fire, and that could be the superposition of the qubit. But I thought they were too big, so I suggested to him microtubules might be the quantum computer that he was looking for, and that his objective reduction, which was the quantum mechanism for consciousness, might be what I was looking for. And so we teamed up and developed a model called orchestrated objective reduction, which is basically shown here, in which each tubulin is not just a bit, but a quantum bit or qubit. Now, we immediately came under attack by many people who didn't like the idea, philosophers, reductionists, physicists, materialists. Pretty much we got skeptical criticism from all corners, and most of which we've dealt with. And I would say that nobody's really come close to refuting us. We've had to deal with some issues. Most recently, there's been a round of attacks by a group of eminent Australians, including this one that said Penrose Hameroff orchestrated objective reduction is not biologically feasible. They also say in the abstract, it's not only not feasible, it cannot be remedied despite any future modification, which is kind of surprising because maybe they're psychic and know that. But anyway, actually their objections are bogus, and I'm not going to go into it now. I just gave a talk. I discussed it at Google as to why they are, and we're writing up the paper now. But suffice to say that it's a bunch of, in my opinion, anyway, it's a bunch of baloney. And if you want, I can go through it later. But that's not really the purpose here. The other attack piece from the same group also attacked us, but it was really more about the Frohlich condensation, and the nature of whether Frohlich coherence is a quantum state like a Bose-Einstein condensate, or whether it's a classical state. And in that paper, they validated that biological excitations discovered by Professor Jiri Pokorny, whom we're going to hear next, they validated his findings of Frohlich coherence, but called it a weak condensation, therefore not really quantum. But from our standpoint, that was perfectly good because we just wanted to synchronize. But it raised the issue of Frohlich condensation. So just final point is that one part of the issue they raised against us is that this cartoon, they said this conformational change requires GTP hydrolysis, and this hydrophobic pocket with aromatic rings with electron flipping, which is our proposal to make the connection to quantum, wasn't valid. And we said, well, it's just a cartoon, and actually this is the latest picture of aromatic rings in tubulin. There's actually 50 of them. And we think that these may mediate quantum pathways through here and leading to topological quantum qubits that Honorbound is going to talk about subsequently. So with that, I will close, and I want to introduce our next speaker, Professor Jiri Pokorny. And while he's coming up, I'll tell you that I've known of his work for many, many years. He is literally and truly a pioneer in the study of electromagnetic fields from living cells, and he's from the Czech Republic. He was a contemporary or a colleague of Frohlich, and I had the pleasure of meeting both of them in was it 95? Well, no, Frohlich was gone by then. I met Frohlich before that, but we met in 95 in Prague, right, at a conference he held, and I was pleased to see that in at least according to our critics, Professor Pokorny confirmed and validated Frohlich condensation even better in microtubules. He found evidence, which he'll tell you about, evidence of coherent megahertz vibration. Now, Frohlich said gigahertz, And he found it at 8 megahertz, slower, but still perfectly fine for our needs. And so I'm pleased to introduce Jiri Pokorny, who will tell us about microtubules as electrical oscillators. Thank you. Jiri, are you ready, or do you want to use the version on my? I'm ready. Okay. Can you use your computer?
Yes.
Okay. While he's setting up, any questions about what I said? Yes.
So you said that tau is important for the structural integrity of the microtubules, right?
Well, that's been the conventional story, that it knits together, and maybe that's true. But the recent findings say that it also acts as a signal, as a traffic light, to tell the motor proteins where to get off. And
I feel like those can't both be the case, because if they were formed for the structural integrity, then they'd have to be like symmetrically dispersed around.
yeah, that's
And wouldn't be able to have these patterns beside it. Because obviously the microtubule is symmetric, it needs to be
I agree with you entirely. I agree with you entirely, and I think the idea that hang on, do you need what kind of connector do you have?
Connector is here.
Yeah.
Normal connector. That one.
Oh, okay, okay. I think tau is found only in neurons. It used to be thought only in axons, but it's also in dendrites. And I'm not sure if it's found in other cells or not, but I'm pretty sure it's not required for the stability, even though that was the story all along. So I think it makes more sense that it's some kind of signaling mechanism like that. And I'm sorry I don't have the reference for that. I can dig it up for somebody else's work about the tau being the traffic signal. But the CAMK2 is very consistent with that. So there's kind of a menagerie of proteins, motor proteins and microtubule-associated proteins associated with the microtubule that can have to do with information processing and cell architecture. Okay, Professor Pokorny, please.
Ladies and gentlemen, I would like to say something about Frohlich Coherence hypothesis, then about our measurements that we performed a long time ago. We started 15 years ago and published in the beginning of the year 2001. And then about mechanism that we assume is important for electromagnetic activity of living cells. Because we assume that electromagnetic activity of living cell is important part of biological activity. First, Frohlich formulated his hypothesis. Formulated his hypothesis in two models. First model is model that describes spectral energy transfer from upper frequency downwards. And the second model is a model of a nonlinear interaction between elastic and polarization fields. This Frohlich never joined these two models into one, but both models characterize Frohlich hypothesis. The first model is based on his work in the beginning of the 50s last century. Interaction of two modes in an oscillating system with a mode with a heat bath, which leads to this interaction between three modes leads to energy transfer downwards the frequency square. The same upwards, but the probability of the up conversion is much less than the probability down conversion. This principle leads to energy channeling towards the lowest frequency, Frohlich assumes that the lowest frequency is coherently excited. It needn't be so. Even the higher frequency modes may be coherently excited, and the lowest frequency mode needn't have the highest energy. And this is similar to a dam along a river. And I would show you what is now known. This is abscissa shows frequency spectrum from acoustic frequency spectrum up to ultraviolet spectrum. And here you can see electromagnetic signal with respect to frequency that were already found. Some of them were this this light blue is only indirectly determined. The other are correctly measured. The highest frequency were measured, or emission of photons were measured by Pop in Germany in the ultraviolet spectrum. Each frequency band has special, very likely special function in biological system. We first, after some considerations, started to measure at about 10 megahertz, and we somehow assume that this spectrum may be important. The measurement were done a long time ago. Now, the second model is based on formulation of nonlinear processes and include in the Lagrangian formulation of kinetic and potential energy with strong nonlinear interactions between electric polar and elastic modes. And after formulation Hamiltonian, after formulation Lagrangian, then that equations of motion were derived, and Frohlich derived special property in this nonlinear system. There are 4 figures in parallel. On abscissa there is electric polarization. Ordinate is modified density of potential energy. And if the parameter U depends on energy of elastic waves, if the energy of elastic waves is small, you have this first curve. It changes if the energy of elastic wave increases, and now appears something special. There is ferroelectric state with polarization vector, and special potential valley is formed. That is the second Frohlich model. These models should be put together. And there are 2 branches of oscillation in this. Just a moment back. There are 2 branches of oscillations in this polarization mode, ferroelectric mode, symmetric around initial point, and symmetry, certain type of symmetry around polarization. I would like to mention that the spectral energy transfer model is based on interaction between elastic field and heat bath. And the ferroelectric state model with potential value is based on polarization, electric polarization field and elastic field. Therefore, these two models should be assumed as full-featured Frohlich system. Pardon?
What exactly is an elastic field?
Elastic oscillations. Elastic oscillations.
Just mechanical vibration.
Mechanical vibrations. And I would like to mention now the criticism of Australian scientists, famous Australian scientists, and what mistakes they did. He used the first Frohlich model with constant coefficients, linearized coefficients. It leads to incorrect results because these coefficients, energy is transferred downwards. The nonlinear mechanism is in this 3-photon interaction. But the coefficients of energy transfer are dependent on excitation. And therefore, there is not the full-featured Frohlich model. They neglected important part, it is nonlinear properties of the system. And now about our experiment. First, you must, I would like to ask you to observe it as something that was started 15 years ago and ended more than 10 years ago with our possibilities at the time. We measured yeast cells and the special system for measurement, this is 10 micrometers wide gold strip. There is a gap between these two gold strips, and the surface is covered by insulation layer. And that window, at that window, the insulation layer is removed. And we measured cells using this system. We use also other system, but this was more perfect. The measurement system contain sensor and preamplifier in double screened box. One screening was from mu metal. Spectrum analyzer, the system controlled by computer. And we also, the biological clock was observed. The cells that we measured in the system to disclose electric parameters were measured using immunofluorescent microscopy. We used this genetically modified system, temperature dependent. The cells at restrictive temperature do not form microtubules, and at restrictive temperature between 15 centigrade and above 25 centigrade permissive temperature, they start to polymerize microtubules. This is synchronized suspension. In the first slide, this beginning is just at the beginning of prophase. 20 minutes late, the mitotic spindle is formed, anaphase after 40 minutes, anaphase A, and after 60 minutes, anaphase B. We also use another system of cells. This is also genetically modified cells, but these cells form or polymerize microtubules at restrictive temperature, but not functional. And after heating to permissive temperature, this non-functional microtubule system is depolymerized, and a new system of microtubules is formed. And here, after 60 minutes, is a fully developed mitotic spindle. The cells are synchronized by the temperature. At restrictive temperature, they stop before M phase because they cannot continue in the M phase. And here is, this picture shows electric measurement in a part of the frequency band. We measured in the frequency band from 8 to 9 megahertz, and this is 200 kilohertz part. The line in this is noise, and this is center part of line of noise, and 6 dB, it is in logarithmic scale, it is not linear scale. 6 dB above is threshold for evaluation of these lines. What's the probability of that? These lines that are higher, that their amplitude is higher than the threshold line, has probability about 10 to the minus 4th. 10 to the minus 4th. And it means that in this frequency band we measured, we may observe one noise line of this amplitude. And but the number of these spectral lines in our measurements that may be assigned to microtubule activity is similar of the same number. Therefore, we evaluated in such a way that we subtracted the observed spectral lines that exceed the threshold. We subtracted from this control experiment number. And we altogether measured about 80 different measurements. And it was the only possibility at the time how to detect all this. And here is 3 figures. The upper part of the figure shows the biological clock, the development of the M phase, the prophase, metaphase, anaphase A, anaphase B, 60 minutes altogether. This is time scale. I will only show this low figure. It sums the upper 2. The ordinate is number of these spectral lines exceeding the level, and subtracted the number of control experiments. We use these type sensors, and all these peaks are statistically significant. Therefore, it corresponds to previous measurement that in the M phase, the activity of cells is the greatest, electric activity. This is average from about 20 experiments. as a control experiment, we used empty sensor, sucrose, sensor filled with sucrose or with non-synchronized cells. And this is, here we use sucrose for control experiment and wild strain of yeast cells. And again, all these 4 peaks are statistically significant. Just a moment. And now for control, the second type of cells, the cells that form microtubule system, but not functional at restricted temperature, and when heated to permissive temperature, depolymerize the system and start to build new system. The upper part, you can see that at the point B, the new system is built, and at point C, the system of functional microtubules are formed. It's quite a different picture from the previous one. Now, I would like to finish this part about our experiment. You must take into account that we measured this more than 10 years ago. 20 years ago, we started which structure may generate the Frohlich oscillations. Which structure? And looking through the cell, we found that we try to find structure that is linear, that is electrically polar, and structure which is supplied by energy. And this structure are formed by microtubules. Actin filaments may also very likely generate a field, but the field is very special or very difficult to determine space structure. And now, what we assume about the mechanism of oscillations. First of all, I must mention mitochondria. Mitochondria are structures that not only produce ATP and GTP, they are multifunctional structure. First of all, during production of ATP, the mitochondria transport protons across inner membrane to intermembrane space, and by diffusion, these protons form a charge around mitochondria. The charge looks like this, it is only model. Mitochondria are aligned around microtubules. And what is important, this space charge layer extends to the distance of micrometers from mitochondria. Here is there are theoretical values of the electric field, static electric field around mitochondria. Abscissa distance, ordinate intensity of the electric field. The electric field at the membrane is 3.5 megavolts per meter. It is extremely high electric field, extremely high. This field was measured by Tyner, Koppelman, Filbert, and published 3 years ago by fluorescent nanoparticles, 30 nanometers in diameter. The dimensions of mitochondria and these fluorescent particles are in size. Electric field is oriented towards in the direction to mitochondria. That's first important point for understanding. We must keep in mind that mitochondria occupy about 20–22% of the cell volume. But this electric field and this space charge layer occupy the remaining part of the rest of the cell. And that is very important to keep in mind. Before, in strong electric field, water is ordered. It is not a normal bulk water what we can get from a water supply. Sources of energy for microtubules. First, growth and shrinkage of microtubules in the interface. Guanosine triphosphate is hydrolyzed and part of the energy is stored in the microtubule. It is, I assume, the smallest part of energy supply. The second part is the motion of motor proteins. Motor proteins move along microtubules, and concerted motor protein action supply energy to vibration states. But again, I assume that it is greater energy supply than hydrolysis of GTP in after polymerization, but that it is also not the main part. The main part, very likely, is energy efflux from mitochondria. Efficiency of production of GTP and GTP and ATP is about 40%. 60% of energy that is supplied to or accepted by mitochondria is released or liberated in the form of photons and heat. Photons, even UV photons, and invisible spectrum too. And this energy, which is called in biological books wasted energy, but it is only non-utilized energy, is partially absorbed by microtubules, and this energy may be the main source of energy for vibrations in microtubules. I would only mention that the total turnover of power in cells is about 10 to the minus 13, 10 to the minus 12 watts, 1/10 of picowatts to 1 picowatt in this region. And therefore, all the energy that I speak about are very small. In the cell, there are two ways of ordering of water. The first one is interfacial ordering. It is ordering around surfaces which are charged. For instance, around microtubules, there is this clear zone or exclusion zone, it is called, about up to 20 nanometers, these zones. And it is proved that this is really organized water. For instance, even heat radiation is small. It looks like black region on the pictures. But it is only partial organization of water. The main organization is produced by the electric field of mitochondria. And this is organization in the whole cell. But we must keep in mind that all the water dipoles are not oriented in the same direction because it depends on the field. For instance, this is interfacial ordering of water around microtubule caused by surface charge of microtubule. It is in the book. This is, and now mechanism of physical processes in living cells. First of all, these physical mechanisms must have energy supply. And energy supply, main energy supply, is provided by mitochondria. Around mitochondria, there is strong static electric field and proton layer. It's difficult to say whether for organization of water is more important static electric field or proton layer, because proton layer, all protons have hydration envelope around them. Therefore, it is both important. And this result is ordered water. Ordered water in the cell. That something that is proved just recently. Zeng and Polak published that paper 4 years ago. And the second important point is creation of nonlinear effects. This strong field shifts all processes in cells into nonlinear region. Therefore, principal nonlinearity is fundamental property of all biological reactions that must be keep in mind these nonlinear effects. And now wasted energy supply, efflux or liberation, and energy supply to mitochondria. Simultaneously, GTP. Mitochondria has also function in apoptosis, but I do not know anything about it. And microtubules, according to my point of view, are the main structure generating electromagnetic field. And I speak about electromagnetic field. They may generate photon and intermediate state and so on. And this electromagnetic field is responsible for directed transport. The upper part is hypothetical. Up to this lower part really has experimental proofs. Directed transport, organization of the cytoskeleton, interactions inside cells and interaction between cells, and information transfer. And now, it is another figure. I would end with this. Thank you for your attention. And I assume that there is prepared a way for Anirban.